Welcome everyone to the new episode of BeyondPhrenology. And today we have with us Dr. Graham Scarr. Dr. Graham Scarr has been an osteopath. Currently he's retired, and he's also fellow of the Royal Society of Biology in UK. I know Dr. Scarr from reading his book, Biotensegrity, which is a very highly recommended book. And he's a biotensegrity expert. I think when we talk about biotensegrity or principles like those in human physiology and movement, two names come to our mind. One is Dr. Stephen Levin, and the second name is Dr. Graham Scarr. So we are kind of very grateful that he took time to come to BeyondPhrenology and discuss all things related to biotensegrity. So welcome, Dr. Scarr, to the show.
Hello, and thank you for that introduction.
So, Dr. Scarr, you are an osteopath living happily in UK, and then you came and adopted and started working on this very controversial idea of biotensegrity.
Yes.
How did that happen?
Well, I've been osteopath for about 15 years or so, and I recognise things in my patients that didn't make sense. When I'd be working on somebody, say I'd be working on the shoulder, and the patient would say, well, I can feel things going on in the right-hand knee. And I thought, how does that work? And these little things kept coming up. And I read the textbooks, and they just explained the standard theory of biomechanics, the musculoskeletal system, levers, and all that, and it didn't quite fit. So I was reading a magazine, an osteopathic magazine, that described a different way of understanding the human body. And the thing that really caught my attention was a picture of a Tensegrity model. It's just like this one here. And all my life I've been interested in natural patterns and shapes, but there were no real explanations just considered as interesting curiosities mainly. So I read this article and I thought, wow, Biotensegrity, that's really interesting. And in the process, I discovered Donald Ingber's Scientific American paper from 1998 called The Architecture of Life. Did a bit of a Google for Stephen Levin. He popped up. I got the same week, I got a flyer for a conference that was taking place in London with Stephen Levin talking, and I went down there and I met him, and it transformed my life. I've been looking at it for the last 20 odd years since. How do you want me to progress with this? I got a few things to
I mean, of course, we want to know what biotensegrity is, what you can show as examples, especially with reference to the human body. Please go ahead and cover all the issues without me prompting you.
Explaining biotensegrity, it's not an easy thing to do. Biotensegrity is a completely different way of understanding the human body and the whole of biology, because it's based on some first principles of physics, and from which everything else is derived. And this is quite different to orthodox explanations that are based on the machine model from Descartes, Borelli, and Newton, which is currently dominated. The mechanistic theories that they promulgated are now well established, but although major developments in computing, mathematics, and engineering have enabled great progress to be made in modeling the human body at every level, it is their analytic convenience rather than anatomical fidelity that ensure their dominance. Biomechanics recognize there are issues and problems, but are generally smoothed over them and carried on regardless.
Right.
Because medical needs drive the enormous amount of research that takes place at every level, with biomechanics co-opting reductionism and engineering as a way of facilitating those goals. And the obvious benefits from such a partnership are undeniable. but the focus on finding quick solutions to real-life issues has not been entirely consistent with the unbiased progression of anatomical knowledge. And it tends to overlook the assumptions and approximations inherent within its methods.
Right.
And they're now routinely accepted as part of the modeling process. So Biotensegrity is essentially an offshoot of Tensegrity. And Tensegrity is a structural principle that describes a relationship between a set of compression elements that are suspended or appear to float within a network of tension elements. So this is the archetypal Tensegrity model. It's called the Tensegrity icosahedron, which I'll explain later. This was discovered in, well, essentially discovered in 1949 by Buckminster Fuller, who was a rather controversial designer, inventor, architect, who was talking about his ideas about that biology could be really useful to understanding new technologies. And he was talking to his student, Kenneth Snelson, who was a young, just in his early 20s. And the student, Snelson, was very impressed with this. And he went and made his own models, which were simpler than this one.
Right.
But Fuller recognized it and then made Tensegrity popular with his geodesic domes that are spreading up all over the place. Geodesic domes are essentially triangulated icosahedra. Icosahedron is a 20-side model.
Yeah.
Yeah, that's an icosahedron.
Right.
And he really promoted Tensegrity in his book, but because his ideas were very out there, very controversial, it got largely pushed to the more esoteric. But engineering did pick it up many years ago, and recognized it something that could be useful. It's just that the analysis of Tensegrity structures using traditional mathematical means is really, really complicated.
Yeah.
But great steps have been put now to make it forward. The thing about a Tensegrity structure is it is very strong, very light in weight, flexible and resilient. It's a truss, essentially, that can change shape with a minimum of effort. It distributes its forces throughout the structure and it balances them completely, and it's intrinsically stable. And a characteristic of Tensegrity structure is that it's non-linear, as opposed to most engineering models, which are essentially linear, or they make approximations to linearity.
So by linear, you mean the relationship between stress and strain?
Between stress and strain, that's what I mean. Yes, that's right.
Yeah.
So buildings and the like are constructed with compression elements. So all the weight is transferred down through the structure to the ground.
Yeah.
Tensegrity models are light in weight. They can remain stable in any position. They're not dependent on gravity, essentially. Biotensegrity is different to Tensegrity in that it's looking at the body from first principles. It recognizes the hierarchical relationship. So each part can be made from smaller parts and smaller parts still. And all I can do now is a quick summary, but we'll work into this over the next half an hour or so. So Biotensegrity has been described in molecules, cells, the connective tissues, its importance to understanding cancer, clinical decision-making, and is going to make a big noise in the near future. It's already making a noise, which will become much more widespread. Once we've got the initial problems with trying to understand one part, picking holes in one part without appreciating the whole, that's been a big issue. Because understanding the whole is a massive job. Biology is just massive. And Stephen Levin started this in the late 1975 onwards. And I've been following along what he's been doing. He's written lots of stuff, but there's just so much more to do. And here we are.
Right,
yeah.
No, he has written quite a fundamental articles. I mean, when I read those first time, while I was doing my PhD. I mean, they
Yeah.
were like eye openers. They give you a completely new lens to look at the world and to look at, the human body. And not just the human body, you can see similarities between, the body and the cell, which no other framework right now gives you. Like
That's
the
right.
biomechanical models we apply right now, the Newtonian models, at the level of the body, they don't work at the level of the cells and tissues. But
Yeah.
this is one of the only models available which is scale independent.
Yes, absolutely. But the problem is that Biotensegrity is enormous. The potential of it's enormous. And Stephen Levin often says things that made me think, what's he talking about? This is crazy. And then I look at it a bit closely, and gradually I start to understand what he's saying. And that's a difficulty, because understanding one bit without understanding the whole, appreciating that hierarchical relationship, or even a heterarchical, where heterarchy is multiple hierarchies all running in different directions. It's not just a top-down hierarchy. The interactions between the body are across every scale, across every part of the system. So indeed, Stephen Levin has been a champion of this all his life, and he's still going as well. But
Right.
I'm starting to pick up some of this.
He very often, puts up these documentaries which are a little cheesy based on their background music, but very informative.
Sorry, say it again.
So he comes up with these 10-minute documentaries, which I've seen some of the podcast, which often have a very cheesy kind of music going in the background,
Yes.
but very informative content.
Yeah,
Yeah.
very entertaining is
Very
video.
entertaining.
They are on YouTube. Do watch them. He's also written lots of papers and several books,
Right.
or chapters in books mainly. So let me just. One of the problems with the musculoskeletal system, that's become the standard thinking about biological movement at the high scale. It's really a simplified interpretation of anatomy. And it's persisted out of tradition, but relegated the associated connective tissues to mere ancillary roles or packing tissue. And that position of the body acting as a machine has become embedded in our thinking. It's a curtailed version of anatomy that's appeared in the work of Vesalius in the 16th century, famous dissector. And it's influenced every clinician and researcher since, which is why overcoming that is a big challenge. So I thought about a different classification for biology, or biomechanics, should we say. We have class 1, which is, what should we say? Class 1 is like a truly accurate representation of the human body. It's the real deal. Class 2 is a temporary approximation of that reality. It's a hypothesis or perspective that can appear to emulate the class 1. And class 3, which is a solution-oriented model that fills some pragmatic needs, such as a robot or prostheses, but differs markedly from the class 1 reality. Because anatomy is really a map. It's a functional map of the reality.
Right.
And just like a map, a geographical map, the map summarizes key places and relationships between different aspects.
No, but it has to be different, because in the body you have to talk about the movement, not in the geographical sense, right?
Yeah.
So we need something more than just the spatial locations and the spatial alignment of the components, if you're talking about the anatomical map.
And of course, if a map is incomplete, then you make another map at a different scale to pick up the details that the first one couldn't do. Right. And that's what the musculoskeletal system is really. It's a map of the body that tends to give the impression that that's it. But it's just not. It's just so much more than that. And The class one's the reality. It's a definition of anatomy has been that the structural basis of the body. But a second definition is the study or representation of that something, of that anatomy. So you've got the reality and the representation of it. And the two are very different. They're fundamentally different. And confusing the two, or putting the two together, considering they're the same, has been half the problem. So the class 2 modeling can pick up some of those details. You can put an approximation, you can put hypothesis there, but it's always an hypothesis. The musculoskeletal system is just a class 2 modeling.
Right. It's very much same in neuroscience as well when we're talking about the brain. A lot of our models, in fact, might go to class 4 and class 5.
Yeah. Perhaps I should make some comment about, I will come back to Biotensegrity, I really will. I want to put some of the problems with the lever system. The body is considered to be a lever system where the bones of structural framework of the body and the muscles move the bones around. And the problem with a characteristic of lever systems is that they generate compressive forces between the sliding joint surfaces. Yet there's a clear space between those joint surfaces, at least in the elbow and knee. That's been demonstrated experimentally. Even Stephen Levin did it with his own knee. He had some surgery on his knee, and he chatted to the surgeon about his ideas, and it was a keyhole surgery, so he's under anaesthetic, and the surgeon agreed to do a few little maneuvers, such as pushing the feet, compressing the feet and the head together, so the knees were compressed. But even so, there was a spacing between the bones. And you could say, well, this is the cartilage, but in between the cartilage between the bones, because cartilage is really soft stuff.
Right.
Which added to the evidence-based. And
which implies almost zero friction, right?
Friction generates shear stresses, generates heat, wear and tear. Bones sliding against each other, even with cartilage on, is not a good idea. And I think the evidence suggests that at least knee cartilage is quite soft, and it's acting more as a shock absorber. It's not just keeping the bones separate. And the fluid in the joint is really, the fluid pressure in the joint is really very minimal. So that's not going to be doing it either. So a lever system cannot passively stabilize itself in the presence of unexpected forces and moments. And it's always been assumed that it's the muscles that do the adjusting to it. But the problem with that is the muscular system controlled by the neural system.
Yeah.
And even the fastest monosynaptic reflex experiences a time delay. So it's likely that unexpected perturbations in the system could happen and cause problems faster than the muscles can react to. And then you get degenerative changes in the tissues.
So that implies that it has to be mechanical, right? If it has to work, the responses to those perturbations have to be mechanically transduced and mechanically responded to.
Yeah, and the thing about the Tensegrity system is it's a body-wide system that applies from molecules, cells, tissues, everything. So if the mechanics of this, if I compress, I don't know how easy that is to see, if I compress top and bottom, the whole system contracts.
Right.
In fact, it contracts auxetically. Auxetically by, I mean, this isn't a Tensegrity. The whole thing expands and contracts. And auxetic behavior has been shown in cells, many tissues. So it's something only been around for about 30 years.
Right.
Auxetic behavior was thought to be impossible, and it's now becoming very, very popular in toys and engineering.
I mean, Donald Ingber's work, over the last 4 decades, I would say, has shown this consistently in tissues and in cells.
Yeah. Donald Ingber's a key player, because he's a person who first provided the empirical evidence for tensegrity in the body back in the early 80s.
Right. I mean, in fact, yeah, sorry.
Sorry, carry on.
I was saying that he has in fact shown that a lot of cellular communication is based on tensegrity properties, or at least tensegrity properties determine a lot of cell fate and cellular communication than a lot of chemical things which we initially thought that might be governing almost every aspect of cell behavior.
Yes, yes. He showed that cells can contract auxetically, and when they change their shape through the cytoskeleton effectively a tensegrity structure of tensioned actin filaments, microfilaments, and compressed microtubules. And it's not a static system. These microfilaments and microtubules, as part of the tensegrity, are changing all the time.
Right.
So any changes, because cells are directly connect to the extracellular matrix around them. Indeed, fibroblast cells produce the collagen and proteoglycans in the extracellular matrix. It enables a transfer of tensional forces from within the cell to the extracellular matrix, or vice versa, from one to the other. And in the process, this process called mechanotransduction causes changes in the molecular behavior in the molecules just within the cell membrane. The signaling cascades that transform the cell from a passive structure to one that differentiates, it can move, it can grow, it can change to different other types of cells. So here's really important. Once you got it, if biology behaved in a different way in the cell, a different way in muscles and bones, that would be rather odd, considering that we've all come through evolution from cells in the first place.
Right.
Why wouldn't the same principles that operated at the molecular cellular level also apply to everything else, which of course biotensegrity explains how that can happen.
Right. In fact, it will be very difficult, because then it opens up this can of worms. For instance, when exactly during development, a new kind of biomechanical systems comes into play, right? When did the transition? So one can ask that when exactly during the development, of a zygote, of a human development, of a baby, when do the real biomechanical, the Newtonian biomechanical principles come into play? Is it like, does it happen like an overnight? Is it an overnight thing? Is it a rough transition thing? Which is very hard to explain, and it's almost intractable.
Well, the basic principles of tensegrity apply throughout the body.
Yeah.
And they're based on, biotensegrity perspective considers them based on geodesic geometry, close packing, and minimal energy. The geodesic geometry, the tensegrity is a geodesic structure because each of the tension and compression elements transfers forces in a linear way, so geodesic. We can also think of the same thing in molecules between attraction and repulsion. Now, atoms, atoms interrelate with each other through van der Waals forces, hydrogen bonding, electrostatics, covalency, steric repulsion, all sorts of different ways that atoms interact with each other, really. But they can all be classified into attraction and repulsion, which is a simplification. But actually, that's what they're all doing. They're attracting or repelling, which is why molecules form as they do. And once you've got that, you've got the tensegrity and the building blocks, for want of a better term, for the rest of biology.
Right.
And tensegrity is close packing because, if you say, the structural elements are as close as they can get to each other. They're close packed. We could, that's an example of close packing in atoms, which form in minerals. They form these close packing shapes. And minerals are relatively static. Molecules, by their nature, are flexible, and they can transform and change. And the most important thing about biology is its energy efficiency. Everything always runs energy always runs to the lowest state of energy possible throughout. And then mechanisms, we feed it with chocolate and pizzas to keep it going, to put the energy level up. But once it's inside, it's always running downhill. Everything inside is running on that trajectory.
Right.
And it's easy to forget those things: geodesic geometry, close packing, and minimal energy. But the same applies to throughout the body. Cells close pack each other. They're transforming and they're growing, they're dying, they're changing into other different cells and then producing things like collagen, proteoglycans, tissues surrounding them. They're always all about geodesic geometry at the basic level. One of the issues with the musculoskeletal system being so dominant and relegated the fascial tissues, the connective tissues, to mere ancillary roles. That's been the current position up until about 20 or 30 years ago, but it wasn't always the case. So in the 18th century, Robert Hooke, the chap with a microscope, looked at cork cells and he wrote a great book called Micrographia. And he had a mate called Marcello Malpighi, who also looked at inside the body with his microscope. And Hooke noticed that the cork had little compartments in it, cells, he called them, after the naming of muscle cell. The cells in monasteries, where monks live in cells, little compartments within a larger structure. And Malpighi recognized this as well within in the body when he did his dissections. And this idea caught on because it was recognized throughout the body. So with their microscopes, they could discern little compartments all over the place, the muscle compartments. And inside the muscles, there were smaller compartments. They didn't know what was inside the compartments. It was just a mushy mess. They didn't know what it did. But they called this the cellular tissue. And cellular tissue had a quite different meaning in the 18th century than it does now.
Right.
And people like von Haller, Lamarck, Winslow, they wrote books about it and really keen on this great cellular tissue and enthusiastic about finding out what they could do with it, what it actually did. It's only later, in the early 19th century, late 18th, early 19th century, that Schwann came up with his nucleated cell theory. Microscopy had improved. They could see smaller cells that actually did things. Now they could see the muscle cells, the liver cells, all the other different cells, which we now call the parenchyma. And of course, this was exciting. Now we had a function for all these bits and pieces, and it became more prominent. And the old idea of cellular tissue faded away, and together with it, the fascial tissues became nothing more than packing tissue. All we get left now is compartment syndrome, which is really poorly understood. But the principle, the idea of cellular compartments in the old sense of the word cellular, is now starting to make a bit of a comeback, and it's currently under the paper that's currently under review, but we'll have to see how that progresses. But the fascial system in general is now receiving a lot of attention with fascial research congresses every couple of years. So,
in fact, there was a recent paper, the fascia being the largest organ in the body, and it got like a lot of press.
It is really important because none of the parenchymal tissues can do anything without the enclosing fascial tissues.
Right.
I mean, the parenchymal cells and fibroblasts produce the fascial tissues. The fascial tissues then enable those parenchymal cells to function. So it's a two-way relationship. And is it chicken or is it egg? They're both at the same time. They're both equally important because neither can really work properly without the other.
Right.
Which means that we can look at bones and muscles in a different way, because bones as because within the compartments of the body, we have the cranium, the thorax compartment, the abdominal compartment, but inside those we have the pericardium surrounding the heart, and hepatic capsule surrounding the liver, the periosteum surrounding bones, the endomysium surrounding the epimysium surrounding muscles. They're all just compartments within this much larger structure.
Right.
This is going to be controversial, and going to hear it for almost the first time. It makes bones to be considered in a different way because they're just specialized regional compartments of fascia. Yes, they both have important things to do because they're different physical characteristics.
You're right about that. And especially if you think from a development perspective, when an embryo is forming and when an embryo is becoming like, the human body with bones, yes, I mean, it's just that those compartments are actually developing a little differently with more, more calcium tissue and much harder. I mean, what else is the difference? I mean, they are a compartment.
Absolutely. And those compartments run throughout the body. And medicine has what's called compartment syndrome. It's when usually groups of muscles contained within a specific fascial compartment within the limbs, for some reason, due to probably a vascular abnormality, the pressure inside the compartment gets bigger. If that pressure isn't released, potential of necrosis and the word's gone from my hand, paralysis. So it's really serious stuff. It can be difficult to diagnose in its early stages, and the only real resolution is with surgery to cut that fascial compartment. When we look at the body in a fascial system as a compartmentalized way, it changes how we find those fascial compartments all over the place. And we can think of cardiac hypertrophy or cardiomegaly, where the heart gets bigger, whatever reason, as a compartment syndrome. I haven't seen it stated as that anywhere. People with fibromyalgia generally have an increased intramuscular pressure. Can we call that a compartment syndrome? So I think this idea, which is really quite new, only a couple of years old, well, since I started writing about it, will have enormous difference to medicine, aside from the biotensegrity issue, which I hope other people will grab onto this, and make a difference to how we recognize, diagnose, and treat many conditions, pathological conditions.
Right.
We haven't mentioned the word biotensegrity for a few minutes, but all this is part of a different way of understanding the human body. It is tensegrity and biotensegrity are the same but different. I think I should mention that people who've been looking at tensegrity in biology have tended to use the term tensegrity out of tradition. That's what the old word, that's what Fuller came up with in the 1950s. And engineering and robotics is using the word tensegrity, and that's fine, that's great. The thing is that biology can do things that engineers can't, and engineering can do things that biology can't. Well, engineering can produce some mechanism to function, to do some particular action, and it takes the stability of the system as a secondary consideration. So many of the things we use tools we use are often quite bulky and heavy in order to counteract the bending forces, the shear stresses that will be inside the material, because metal's quite strong stuff. So it can do things that biology can't do. But there was a classification introduced, an engineering classification on tensegrity introduced in about 2001, I think, by Robert Skelton and et al. And they chose a slightly different approach. So with Fuller's definition of tensegrity, the compression elements always remain distinct. But from an engineering perspective, that doesn't necessarily need to be the case. So they'd have joints where two compression elements would join together, or three or four, and they would call them class 1, 2, 3, and 4.
Right. It's a beautiful book. I've read the book. Yeah, they have a book as well. Yeah.
Yeah. From a biological perspective, that doesn't make sense because only
Right.
class 1 is a tensegrity.
Right.
Stephen Levin recognized this, and because he didn't want to avoid the confusion between the two, he introduced the term biotensegrity to make this separation, which is why biotensegrity is now, I would say, the word of choice. But of course, many people have published great work, Ingber included, considering tensegrity. So when you read the papers, the biological inspired tensegrity, read it for what it is, and essentially meaning the same as biotensegrity. But the two terms are not necessarily synonymous. It depends on the context. So I just make that as a sideline, really. When it comes to modeling tensegrity from a biotensegrity perspective, we then have to match the engineering technical side with the biology side. And this model here, which is now about 15 years old, a bit battered, which is a tensegrity model of the cranial vault. The cranial vault is the box type of the head where you've got the face at the front and the base underneath. And the cranial vault consists of several bones. And while standard line is that the bones are fused in later life after the age of 20 years, and that's it.
Yeah.
That's a bit of a simplification, because some of those sutures between the bones, the fibrocartilage, they can remain patent throughout the whole of life, or they can have a thin veneer over the surface, on the outer surface, but inside they remain quite patent. So there's a huge variation. Most animals, many animals, retain full patency between the bones throughout the whole of their life. So a biotensegrity model of the cranial bolt suggests that because of the particular alignment of the bones, because they have curves, the underlying dural membrane, which is tensioned, pulls the bones together. But because of the particular alignment, which I can't show on that, but the original model, those bones didn't touch each other. They all remain quite distinct. Something like that, it means that the growing brain doesn't need to provide all the force to push the bones outwards. Indeed, that was one of the big problems with that idea, because they couldn't explain how this little force could produce such a big change.
Right.
So there's one example here.
So it solves a developmental conundrum, right? That how does the skull actually expands in a growing baby?
That's right. Yes. Yeah. Of course, a young baby's none of the bones are fused together. There's space in them, or they shouldn't be fused. They have a spacing between them. I think a different understanding from a biotensegrity perspective would change how we think about those bones. Of course, it's not bones; they've got cells inside them. They've got molecules inside those. Thinking of that hierarchical structure, I think it could change how we think about, it'll change treatments of the body. And a year ago, I coupled up with some people in Canada, and we produced a tensegrity model of the cranial vault on a computer through a simulation, showing how the system could remain completely stable, and yet all these bones could transform themselves. And we've yet to write it up into a full paper, but it's available on the internet to at least see the abstract and what we propose. So I think there's great possibilities, but they're all just possibilities, little ideas coming forward.
I mean, there are very few people right now in the community who have adopted this model. So once we have more, especially the ones who have the technical expertise, we'll see more and more coming. I mean, I've read your papers on the tensegrity model of the arm, the tensegrity model of the knee, the elbow joint, and they're beautiful. But yet we have other people to pick up those models,
absolutely,
and refine them and develop them further.
Yeah, the same with Donald Ingber's work with the cells and mechanotransduction.
Yeah,
he's done a lot of work. A lot of people are following that up. But there are also people who pick holes in it. And a recent paper by Bogdadi et al., I think last year, sort of picked up the momentum of all that research, collated it all together, and I think it'll make a big difference. So I'm a champion of Biotensegrity, but because the subject is just so huge, a lot of what comes out of this has to come from other people with their own specializations.
Right.
Let's see. Giovanni, you had several ideas you wanted to.
Yeah, I would really love, if you can go through a little bit about the Biotensegrity models of the spine, for instance, or like dinosaurs' neck.
Yeah, yes.
Yeah.
Yes, I do need reminding these things. Yeah, the spine traditionally is thought of as a column of bones, like a stack of bricks. The same way as building, a stack of bricks that
Right.
held together. But the thing about the spine is that it's flexible. I mean, there's lots of muscles, fascial tissues. It can bend and distort and do all sorts of things. If we consider the spine as a purely compression element, then you have to ask, what about horses and most animals that have a horizontal spine? We can turn upside down. The spine works perfectly normally. Animals don't need a compressive spine. Why would we? If the Tensegrity system, which is so enormously complex that we can't really, it's very difficult to model. I got myself two vertebrae, put them together with a Tensegrity thing, and the two are held apart. But that was just a simplified model.
Right.
But if we look at the spine as a flexible structure, I think it will change ideas about idiopathic scoliosis. Who knows why the spine distorts? It does. Forces of tension and compression acting at different size scales is the key to understanding these things.
Right. It also explains, the idea that if the spine is a completely compression element, then it's just unsurmountable thinking that the lumbar spine would be under so much of stress all your life. But that's
right.
actually not the case.
Yes. And it's not a fixed system. The cells inside the bones, within the muscles, within the fascial tissue, those cells are changing all the time, turning over. If it was a purely compression system, it would just, I can't conceive how it could possibly happen. Of course, just because we can't conceive how it happened doesn't mean it's Tensegrity. But currently, Tensegrity is really the best model to explain all the possibilities of these things.
Right.
And the same with the spine connects to the cranial vault, connects to the cranium, to the pelvis below. The pelvis was always thought to have a sacral keystone like a bridge with a keystone on top that's pushing down, and in between these pelvic ilia. It doesn't make sense because of the compression there, and the keystone be falling out of it anyway because the pelvis is at an angle. The Biotensegrity model transforms that. Chap Tom Flemons, who died a couple of years ago, did a lot of models, very good full body models, some of them life size, with all the joints models of Tensegrity. Of course, they were gross simplifications of the reality, but they got the idea across of what's possible. And he showed that the pelvis can exist quite happily as Tensegrity, even when it's compressed from above. And the same thing goes for all the joints. The feet, for example, hugely complex structures, and yet we can run over them, run over rough ground with ease. And if the brain had to process that, or even just spinal reflexes had to process all that information, it would be enormous. It wouldn't be able to do it in time. And yet running fast over rough ground is commonplace. People climb up mountains and do all sorts of things like that with ease.
I mean, we can't model those forces, to be honest, using the best, most advanced computers right now, because you don't understand the relationship. Yeah. And assuming that brain computes, and then second, assuming that brain computes that particular interaction is just a wishful thinking.
Yes. Yeah. But the thing with Biotensegrity, we can't prove it. Biotensegrity is a concept, really a concept. It's a way of thinking out the body. I don't think you can prove it as such. What'll establish is that so many people find it so useful for different modeling approaches and doing things.
I think you can. I think you can do that. So if you look at Donald Lingberg, his work, he starts with the biotensegrity axiom or tensegrity axiom in his case.
Yeah.
And then he comes up with predictions, and then he sees whether that's what he tests, in his experimentation, whether the cells are responding in ways which is consistent with the tensegrity model or with the Newtonian model. And what he finds is that they are responding in ways which are consistent with the tensegrity hypothesis.
Yeah.
So I think there are still ways to prove it. There are ways to prove the predictions which follow from the biotensegrity axiom. So it can remain within the idea of hypothesis-driven science.
Yeah.
Unlike a lot of criticism it often gains. Yeah.
Yes. Yeah, you're absolutely right. And I think it's Ingber and Levin, particularly, they were able to make predictions, understanding that the whole body worked in the same way. They weren't just, there was, more than just the one little bit. They had the vision to see that. And as you say, people are picking it up on it.
Like for instance, when we make a building with all the compression elements, we tend to have, like, the pillars, for instance, tend to be thicker at the bottom. And then as you build up, we make those pillars thinner to save the material, right? But that's not the case, for instance, in the spine. The load on the lumbar spine is, according to the compression model, the Newtonian model, would be so much higher than those at the spines in the cervical region. But we do not see change in material density or change in the girth of spine as much as the Newtonian model would predict.
Yeah. I think so much my work is not, well, it isn't empirical research, but it's thinking about it. It's pulling the information. There's just so much information out there in the literature to support the biotensegrity model. Just so much. It's a matter of, often it's hidden away in obscure journals, or hasn't been put together with this bit, with that bit and that bit. And it's really my interest, my focus, to pull all those bits together, which is why I wrote the book in 2014 and again in second edition in 2018, which is now 5 years old and needs drastically updating. But to pull all this stuff together and present it like we're doing here and other places too. Because biotensegrity really examines the complexity of the human body and the whole of biology. And this is a system that's evolved over hundreds of millions of years. Biology always does things the most inefficient way, energy efficient way, but it's evolved that way because it's the most efficient way of doing things. And now understanding that is an important thing to progress on that. One of the things I haven't talked about is closed kinematic chains. As an alternate view of the musculoskeletal system, Stephen Vogel in 2013 wrote a book called Comparative Biomechanics, a very interesting book looking at biomechanics throughout the whole body. But he specifically stated in this that closed kinematic chains have no real relevance to the human body, but they occur in lots of different animals. And that was a really unusual thing to say. And I looked in the literature of that, and closed kinematic chains, which
are all over the place. The body is a closed kinematic chain instead of open kinematic.
Absolutely.
Yeah.
So that's a 4-bar. It's a simplified closed kinematic chain where each bar connects to all the others and influences position of all the others.
Yeah.
Tensegrity is inherently closed kinematic chains. You have triangle, they close. The bits between, if you can see it, the 4 bars which expand and contract.
Right.
They're four
No,
parts.
it's pretty obvious. I mean, when you look at the leg, for instance, and you consider the muscles as well as the bones together, they're apparently closed kinematic chains.
Yeah.
It's only when you're looking just at the leg that the leg seems to be open kinematic, in the world. But
Yes.
In a little higher resolution, and it's a closed kinematic chain.
Absolutely. This is one of my first models of an arm as a tensegrity, very primitive, fallen to bits now. The arm, the hand here, is full of closed kinematic chains. And of course, the closed kinematic chains have always been there. It's just the musculoskeletal muscles move bones doctrine has predominated.
Right.
Thing about closed kinematic chains is that, with few exceptions, their behavior, mechanical behavior, is non-linear. The mechanical behavior of living tissues is non-linear. So the two tie up: closed kinematic chains, tensegrity, biology. And in fish jaws, I mean, that's been going for 50 or more years, research on fish jaws behavior, the way the closed kinematic chains enable the jaw to open and close.
I mean, Mantis shrimp, hammering in Mantis shrimp,
get much faster than muscles can contract. That's a very efficient system. If the system itself, if the structural system itself can be involved in the movement in a controlled way,
right?
Tensegrity structures are controlled. That motion is controlled within the structure itself. It's just me putting the extra energy in to make it do that. And about 20 years ago, Robert Connolly is a mathematician, university in US, I forget which one. He published a paper on tensegrity and mathematics, which I read about 10 years later, thought, wow, this is really important stuff, but didn't understand a word of it because I'm not a mathematician. It's only 3 or 4 years ago, a friend of mine, he explained what was in that paper, and it suddenly opened my mind to what was possible. He described something called superstability, which is a mathematical concept, and Madhur can probably explain this better than me, but essentially it's looking at tensegrity. So you have just comparing it with edges. No, I'm not going to even just start there. A super stable tensegrity structure can change shape, transform itself from one state to another, which can explain how a living organism can grow, divide, differentiate, repair itself, move, all with a minimum of effort. And I think when someone's written a better description of superstability in biology than I have, then that will become an important thing, because it can be analysed mathematically and with engineering as well, I guess. So I think that's a big thing for the future.
I mean, it involves very different kind of maths. So I was trying to understand myself a lot of tensegrity maths, especially the transmission of forces from one part to the other. Because, for instance, I mean, we all know that NASA has been building tensegrity robots now, and a lot of new development in robotics is focused on tensegrity kind of models of robots, especially because they're unidirectional. You don't, they're omnidirectional, so you don't care, how they land, how they navigate, and also you can put payloads in them, and the payloads can be launched pretty safely compared to, another Newtonian packaging. And it's not easy because forces are so global in a tensegrity system that modeling them becomes a little harder. So you need a lot of graph theory, for instance, and it goes into this multi-dimensional, multi-body kind of ideas, and computationally expensive to model. So not an easy thing.
But the interest is there. That interest is there, as you say.
Yeah.
I mean, as you say, robotics, space research is a big thing. And biology, the biotensegrity aspect, did have a feed into that, an inspiration to that. And the same way, robotics and space research generates ideas and ways of thinking that stimulate thoughts in the biologist as well. As well, that's useful, that's interesting, because mechanics, engineering mechanics, it's really based on the feasibility of the structure, the material properties, the cost. It has a different focus, but biology can influence that, as it already has done. And the models they produce in robotics can have, of course, a medical impact, because it comes back to improve things in medicine, but also stimulates ideas in biology. So Biotensegrity isn't completely separate from everything else. It's all these things merge into each other in different ways. It's working that out, which is half the fun.
No, it talks to some fundamental issues, like, for instance, we can sleep upside down. We can sleep on the tummy, or we can sleep on the back, for instance, right? And we are completely fine. I mean, again, that's not the case with the Newtonian system. A Newtonian system is optimized for a certain direction.
Yes.
Right? And had it been the case with us, we would have definitely been sleeping just one side, for instance. Or it becomes even more important for a developing infant. The growth would be so much hindered by sleeping on one side compared to the other, had it been a Newtonian system. It's only because it's a Tensegrity system that the growth progresses unhampered, no matter the direction of sleep.
Yeah. People often do talks or have done talks to therapeutic people, manual therapists, osteopaths, physiotherapists, massage therapists, and the like. One of the commonest questions is, how can we use Biotensegrity in treatment?
Yeah.
I've always had to scratch my head and think, I know when I was practicing, thinking of this body in a different way, rather than just muscles moving bones, thinking of the whole thing is interrelated to each other as part of osteopathic training anyway, but I didn't understand it fully. But Biotensegrity made me think how the body works in a different way. Actually verbalizing that into how you do that is not always so easy, because it just changes the way you think about it. If you think about the body differently to the standard way, I think you're going to perform different sort of treatments in a better way. And that will become more specified as we move on, I'm sure, no doubt.
No, what I find interesting is that in 2023, when we think that we are so advanced and we know so much, as soon as you go to the Biotensegrity model, you suddenly start realizing how little you understand about the body, like how little. I mean, we have just figured out that probably this is the way to think. I mean, the content is still not there, right?
It's a shock and very disconcerting. If you spent your whole life in one view, it's rather, can be not very nice to think, oh, there might be something better. But everything takes time. And Tensegrity, Biotensegrity are now becoming hot topics in many fields. So I think that's the way forward, and lots of things will come out of that, spill out of it. The compartments, the professional compartments thing I was oblivious to that 2 years ago. I'd had ideas about it, read little bits here and there, but it didn't really make sense. And now it's starting to make a lot more sense, and I think other people are picking that up.
No, if you talk about that to a developmental biologist, I mean, they would completely sympathize with it. But they'll say, like, yes, it has to be compartment, because when you look at how an embryo grows and and becomes it would be much more apparent, for instance, when we can see things. So human development is not very visible, but the development of a lot of systems, like, for example, chicken, you can scan the egg and you can see the development in real time much more easily. So you would see that how that compartmental happens, because it's not that bone become bones in just one day. The same compartment is something else, and that it starts accumulating calcium and more it becomes more calcious over time. So it has to be a compartmental idea. I mean, beyond doubt.
Yes, and cancer research, as we're picking up on the fascial system for the last 20 years, 10 to 20 years, in a much more bigger way, because they recognize that the cells, the fibroblasts, cancer cells, the extracellular matrix are all intertwined. Each one affects the other. And it's only a matter of extension from that into the fascial system and the whole body. So there's a lot of work, as you say. Technology is advancing all the time. People's thinking is advancing all the time. And it will transform the way we think about ourselves and the body in general and biology in general. So what I've presented is can seem a bit ragged, a bit here, a bit there, but that's the nature of life. It's all sorts of little bits that form together. They form together to form a complete whole. And it's the whole we need to be looking at, not just the little bits. The little bits have to be understood in the context of the whole. I think that's the biggest difference that will happen. Everyone knows that, but in reality, it's very tempting to go down a rabbit hole and explore little bits on their own.
And that happens in the biotensegrity model, because the same element which is under compression, or which seems to be under compression at some scale, as soon as you like zoom out, the same element becomes more like an element of tension rather than compression.
That's right. Yes, tension and compression are inseparable.
Yeah,
they can't exist without each other. So what? I'm not sure what to say more now.
So yeah, a little bit more about how's your work going along with the I saw you're a part of a Tensegrity TEAT group. So what are you doing with that group right now? How, in which directions are they progressing? And are you seeing more kind of more volunteering, or I would say kind of a religious conversion of existing biologists
No.
or clinicians into biotensegrity?
Well, one of the biggest problems is getting the word out there.
Yeah.
Unfortunately, I mean, Steve Levin's done a lot to promote it and has a huge number of people, particularly in the therapy world, manual therapy world, and surgery as well, following what he's interested in, what he's been talking about.
Right.
But there are also people who violently, vehemently dislike it for whatever reasons. And between the group I'm working with Steve Levin and other people, we've written papers. Trying to get them published is not so easy.
Yeah.
Because it challenges conventional thinking. We have had several papers published, but there is a couple of really important things, but I'm biased, that are struggling. They're not there yet. And I think because it's a matter of the tide of tradition has to be overcome. But we keep trying different ways, putting in different things. That's the way for the future.
But do you have more young people, people who are starting their career? Because if you're like a 50-year-old professor in general, you have multiple grants and your lab is working on a concept or based on a paradigm. It's hard to imagine that they would change, right? And they would ever agree that they were wrong or the model was incomplete. But for a new person, just coming into science or starting their new labs, for them it's much more beneficial to adopt like a new model where they can carve out their own professional space as well. Not just from the science perspective, but also from a sociological perspective. So I would assume that for younger people like me, it's a more both attractive as well as might be a lucrative way to shape research, right? So do you see some? Yeah.
There are several PhD students working on different aspects of this right now, and some of them have graduated and now going into labs, so they put that input in, even if they only mention it. And but it's all a bit different. We don't know where everybody is. A couple of years ago, when the coronavirus was around and in the UK, which is where I am, we had lockdown, so nobody could see anybody else. We organised a what we call the Biotensegrity Tea Party, which was an open source Zoom meetings, very informal.
Yeah.
We would meet on a Friday afternoon, put them on YouTube, where people would talk about different aspects, some of them closely related to Biotensegrity, some on the fringes a bit. But it generated a big audience every week for 18 months. And then, of course, we all went back to work, and we still organise those on the Biotensegrity archive website and YouTube.
Right. I'll put those links. I've seen some of those episodes, and like really, really phenomenal discussions. In fact, I have been taking notes for the last couple of weeks, and a lot of research ideas. I mean, if you listen to them thoroughly, you can come up with these ideas and test them in the lab, and probably like publish really impactful research from out of those discussions.
Great. Yeah, that's great. People who can do the actual research, because for myself, I don't have access to that thing, but people like you, like yourself, right, and others can get in and show certain things which didn't understand before. That's great. I think that's going to expand more. So what you're doing is definitely on the train of that.
Yeah, so far I have been more from the statistical standpoint. I've been using Biotensegrity more as explanatory for the kind of statistical results which we see, and they align much more with the Biotensegrity, the idea of nested hierarchies in the Biotensegrity. But I have been trying for some time now. I just started my new faculty position to get a project funded for actually testing the Biotensegrity models in the lab, like what kind of movement outputs Biotensegrity models would generate compared to Newtonian models. So far, I have not been successful with funding and to get a student to work on, but that's in the process. Hopefully.
Yeah, it'll come. It'll happen.
Yeah,
it's going to happen because so many people in the scientific world know the word tensegrity, Biotensegrity, and naturally many of them are curious. So
yeah,
I think when NASA sends a rocket up with a Tensegrity robot, that'll make a big difference to everyone will know the word then.
That will definitely make a big difference. Yeah. And it will also make like a really beautiful first paragraph of the grant proposal.
Yeah. Yes. And I've never done a grant proposal because I'm always been on the therapy side of things, but right. I know they can be challenging.
But I guess you might have a much higher intake of these ideas in the therapy world, right? With physical therapists, for instance.
Much more what? I missed that.
I think these ideas would have made a better way among physical therapists because they are practicing, so they want something better to practice. I mean, they are not into one way or the other of modeling the body. That's
right. Yes, because with physical therapists or hands-on therapists, they're touching the body every day, multiple people often.
Yeah.
And when you're doing that, you pick up things that no general hospital practitioner might pick up because they don't have the time for that particular thing. But hands-on practitioners, you can feel all sorts of things going on. Just the explanations of how and why they do like that are sometimes questionable, but there are definitely things that you can feel in the body that are not in the standard textbook yet. And I think they will. And I think Biotensegrity is a big part of explaining some of those things.
Right.
We'll see.
So, Dr. Graham, what resources do you recommend the viewers who are interested in Biotensegrity to read more and to learn more about the idea and how they can, use it to shape their career trajectories?
Okay, the basics of Biotensegrity, of course, my own book, Biotensegrity, the structural basis of life, which is out produced by Handspring now, taken over by Hodder and Stoughton. That was in 2018. As I say, it's 5 years old. If I did it again, it'd be quite different, but it's a start. Other people, John Keeley, who's a sports researcher in Dublin, he wrote excellent paper in 2016 called The Uniqueness of Human Running Coordination in Frontiers of Psychology. He also wrote another paper with Jan Wilson called The Multifunctional Foot in Athletic Movement, Extraordinary Feats, FEATS, by Our Extraordinary Feet, FEDT. Extraordinary Feats by Our Extraordinary Feet.
Beautiful title, yeah.
Bogdadi in 2021 wrote Revisiting Tissue Tensegrity, Biomaterial-Based Approaches to Measure Forces Across Length Scales, an excellent review, particularly at the cellular and extracellular matrix level. We also have Turvey and Fonseca in 2014.
Right, the landmark article on haptic perception.
That's it. Stephen Levin's article, I think the best one is Tensegrity, the New Biomechanics, published in a textbook of musculoskeletal medicine, published by Oxford. It's a chapter in the book. He's written lots of things you can find on his website, biotensegrity.com. I think the one chapter in that book, Tensegrity, the New Biomechanics, you can find it on Google.
Yeah, I think the PDF is available online. Yes, I'll put a link. Right. And of course, you have several articles on different joints, on the modeling of different joints as Biotensegrity systems. I'll put those links in the introduction as well.
Yes, on the cranium, the elbow.
Yes.
All sorts of other things. I got a whole mess of models here I was going to show, but that's all right.
No, why not? Yeah.
A bicycle wheel is a tensegrity structure, not your usual one.
Right.
But it's, if you think of the rim of the bicycle wheel as a compression element, and the hub, it's a compression element suspended within the spokes. Of course, compression element, it's a different take on the standard model of this, but it's same principles, tension and compression. We got the famous 1960s models, popular in 1960s, of tensegrity.
Yeah.
You combine tensegrity models together, produce an arch, a chain, and those kinematic chains. This one here, those kinematic chains on, I don't know if you can see that.
Yeah.
Just connected, in this case, triangles and 4 bars, showing that the structure itself controls motion. This is another tensegrity model. None of the struts actually touch each other. They're all slightly suspended within the compression element. Well, this is like a nanostructure they use.
So are these structures made by yourself?
These are made by myself, yes. There are an infinite number of different tensegrity possibilities, which means that once you start making models, you need never stop, because there's so many different avenues to. I have hundreds and hundreds of them.
And that's the box of sticks you have for these models, right? On the top, it says sticks behind you.
Sorry, I missed that.
The box behind you says sticks.
Sticks.
And I guess those sticks are for the tensegrity models.
Yes, when I go to conferences, I do workshops often and take a supply of little kits that I get people to make. So those are the ones ready to take to your next workshop. I have nothing planned at the moment.
Right. So, Dr. Graham, also, do you know of any resources? Like, are there companies who are making these tensegrity models as well, like more complicated ones? For teaching purposes, or everyone is making their own models like you do?
There are a few. Yes. I can't think of their names now. Eli Thompson is one person. I can't remember his business name. He's in the US.
But if you can send me those links, I can put those links in the introduction as well.
I'll find them and send them to you, yes, because there are people making them. The best way, once you've got a model, make your own. They seem like really complex, but they're actually very easy once you know how. And once you've got a model in your hand and can play with it, even if you make your own, even better, you understand the structure so much better. But buy one if you got. Contact me, I'll send you a kit. I'm quite happy to do that. I do have on my website, not my website, on YouTube, I have a video showing me making one of these out of a single string. So rather than, there's 24 strings in this,
Right.
which is very takes a long time, but making one with a single string takes about 5 minutes.
Right.
Once you master it,
Is also making the same model with multiple strings. Every time you put a knot, the length of the string kind of changes. But with one string, you can kind of adjust the strut so that you can still make adjustments within that string.
Yes,
Yeah.
you can
That's much
change.
easier.
This one can't change, but yeah, there are so many possibilities with Tensegrity models, which will stimulate new ideas. Lego firm a couple of years ago had a competition who could produce the best Lego Tensegrity, and they were very impressive. So look up Lego Tensegrity.
Oh, so they do have Tensegrity models or Tensegrity kit.
Yeah, I'm not sure they'll do kits.
Okay.
But you can see what's possible there.
Okay. Oh, I guess based on existing Lego models, right? Existing Lego kit.
They're what?
So the Tensegrity model you're saying in Lego were made of existing Lego pieces.
Made of Lego, but obviously have strings between them as well.
I see.
To keep separate bits. But yes, the haven't got the model here, but it's looked like the one bit floats on top of the other in the air. And
oh think, yeah, the famous one. Yeah, I know those ones. A lot now a lot of hobbyists, kind of enthusiasts have been making Tensegrity furniture right these days. They're not like pure Tensegrity in this sense, the two Tensegrities, but they adopt Tensegrity principles to make tables and stools and pieces like that.
Yes,
right.
And it was said in a recent publication, I won't name them, that the model I made for the Tensegrity Vault, initially I started with curved struts, bits of aluminium, and it was stated that can't be a proper Tensegrity because the struts are curved. From a Biotensegrity perspective, that doesn't matter at all because each structure is a hierarchical, so you can have a curved structure. The principle of Tensegrity is compression elements floating in a sea of tension, which is fullest definition. Of course, engineering can make modifications which are fine for that purpose, but it is a Tensegrity, even with curved struts.
Right. I mean, in that case, biology has curves all over the place. I mean, it hardly has straight lines the way, engineering has. But
it all comes down to the atomic level where the attraction and repulsion, those forces always interact in straight lines. The closest distance between 2 points. That's what it all is, which is invisible geodesic, but that's where molecules are. There's no strings between molecules; they're invisible.
Right. No, it's a beautiful idea and a beautiful framework. And for the viewers, I really recommend delving deep into Tensegrity. I remember I discovered this concept while I was doing my PhD, and for just like weeks and weeks, I was reading about Tensegrity, and I took a chance and I went to Society for Integrative and Comparative Biology. I gave a whole presentation of Tensegrity. To my disappointment, I had about 10 people in the room because not many people had heard the term, so not many people came to that session. I thought I did a good job with my presentation at that point, but yeah, it was very challenging to put the idea, in the front perspective. But recently, I've met a lot of practicing biomechanicists who are actually who do see value in that. But however, they also see that it's challenging for them to adapt that model. They would rather want somebody else to do some more work and then it's like who would be the guinea pig.
Yes, that's right.
So I think that's where we are with Tensegrity. Like some people have to take the risk and do some breakthrough work, and that's when we will see a lot more adaptation, at least in the more formal academic community.
Yeah. And the thing about when you try to explain Biotensegrity, where do you start? Because you can't understand one bit without understanding all this, and can't understand this until you've got all that. So that's always a challenge. But
no, that's a challenge with most things which are, I won't say complicated, most things which are fundamental or more holistic. or more encompassing here.
Great.
So thank you, Dr. Scarr. That was a wonderful conversation, and I hope we have created a research which will stick for time and will guide a lot of young researchers for long.
Yeah.
And I hope to have you back again, discuss a couple of, I mean, there are a lot more details, about Biotensegrity, which we have not discussed, we haven't gone through. A lot of them would also need you to have a presentation, I guess. It's hard to speak without, without having an illustration or, details on the side.
Yes. I hope
so
we got the point across.
Right.
My pleasure.
Yeah. And one of the topics which I really wish we would have covered, but definitely we'll do it next time. It's so much more detailed, is the center of mass. So have a great day ahead, Dr. Scarr.
Well, you too.
Yeah.
Thank you.
Bye-bye.
Thank you all.